going to be delivered by Professor Jin Ho. He's currently the Provost Chair, Professor, Director of the N-of-1 Institute of Health, Director of the Institute for Digital Medicine, and the Head of Department of Biomedical Engineering at NUS. Now, Professor Ho's work, I think, looks into leveraging on digital medicine to optimize human treatment for broad indication ranging from oncology all the way to infectious diseases. So without further ado, let me just let Professor Ho take the floor, please.
Thank you so much for the kind invitation, and I wanted to send my greetings to everybody out there today for attending. So the talk today will focus on how we are leveraging various digital medicine approaches to impact patient care. And if we look at the bottom here, I'm going to be highlighting work that really spans across different institutes, different departments, and really across the campus, engineering, medicine, and well beyond that. As it was mentioned, I currently direct N-of-1, which stands for N-of-1, which means we personalize clinical trials to all the patients that we treat. Also, the Institute for Digital Medicine out of the Yong Lin School of Medicine, also known as WisDM, and the departments of Biomedical Engineering and Pharmacology. So it really is a multidisciplinary effort. And today I'm going to kind of take everybody through a couple examples about what we do and use some actual patient use cases to demonstrate the impact I think we're making in trying to think a little bit differently about how patient care can be personalized. I did want to start off by acknowledging the team that makes this all happen. And this is really possible through substantial interdisciplinary interaction. And as expected, we work with a team, a community of amazing clinicians from oncology to neurology. But we also work with physiologists, bioengineers, electrical engineers, cognitive psychologists, and beyond, including the business school, right? And beyond beyond startups, as an example, which there are amazing resources here to pursue that. We also work with NUS business to look at things like operations, healthcare economics, and beyond to ensure that we can carry home the ethos of the institutes, which is technology alone cannot transform healthcare, right? And so whatever we do, we want to ensure that we are interacting with the right stakeholders to advance beyond ideation and validation and move towards scalability and deployment. And part of that includes areas like healthcare economics, behavioral sciences, and beyond. And so I'm going to start off by looking at some challenges and how we are overcoming those challenges. I'm going to start off with a major challenge, which is the cost of bringing a new drug forward is substantial. It's billions of dollars. And this is from a few years ago, and these costs have gone up. And so why? Why is it so expensive to advance therapies to patients? And what we know is it takes a long time to bring drugs from development through approval. Success rates are low. And especially when we look at combination therapy, the process of resolving which drugs to combine and what respective doses to give each drug is a complex process. And here's a challenging one. Even good drugs given at the wrong dose can equal no efficacy. All right, I'm going to repeat that. So even if drugs could be effective, but you combine them together at the wrong dose, there could be no efficacy at all. And beyond no efficacy, this creates the misperception that these drugs cannot be effective for that patient when in fact they can be effective for the patient. So there's a lot of information we have to resolve in order to truly optimize and personalize care. And so I published this paper in 2020, AI for Cancer Therapy and Science, which looks at this roadmap for advancing therapies with AI. And now, when we think about AI for drug development, we often think about the discovery phase, right? So we think about target ID, lead generation, and optimization. But I wanted to highlight that the work that we're doing actually focuses across this spectrum. There's a lot of great work happening in the discovery phase, designing new molecules and beyond. But how we steward those molecules forward is critical. How we combine, how patients are matched to trials, and ultimately how these are dosed, right? So we have to think beyond just developing good compounds and think beyond big data. We have to think about how do we take these promising compounds and use them optimally. We address this using approach that we say are all parabolas, right? And so when we see here, this is actual patient data of dose optimization for HIV patients all the way to transplant patients, and how we can find that optimal way to both select the right therapies, but also select the right doses of each therapy, because there's such an intercorrelated sets of information. So I'm going to give some use cases here. I'm going to start off with liver transplant. So this is a trial that we ran out of UCLA a few years ago. And this is actual data, dynamic data of a liver transplant patient. Now, what happens when a patient comes in with a liver transplant is after the organ is transplanted, we have to ensure that immunosuppression dosing is optimized, because you can either create neurotoxicity by overdosing the immunosuppressant, or the organ can reject if you underdose. And what you're looking at here is week 1, week 2, week 3, and week 4 for a patient. You can see the interaction of the drugs in this patient and how dynamic it is. Now, for transplant, the dynamic nature of patient response is well established. There's a lot of titration and kind of decrease the dose, increase the dose to try to get the right level of the drug in the blood. But using the study, and this is not modeled, this is not computational, this is actual patient change in time, we were able to optimize that dose ranging dynamically. And the outcomes of this trial were interesting. So the curate arm, where we were able to harness our platform to optimize dosing, they were discharged nearly a month earlier on average compared to the control arm. And in every other metric, time within range, time out of range, et cetera, the curate arm outperformed the control in every single metric. The time to discharge is not a standard outcome measure for transplant studies, but this was a first in human study. And what was important to note is that in the US, healthcare costs are substantial, and getting patients out of ICUs faster can certainly improve outcomes potentially and decrease cost of care. But more importantly, these are immunosuppressed patients that are being removed from the potential of hospital or community acquired infection. And so this is an important study. And what you're looking at here is a snapshot of different patients on the study, and you can just see how different they are from each other in terms of how their drugs are interacting. We're going to look at a second use case. This is an advanced prostate cancer patient that was brought to us, and this patient was being given 2 drugs at the time. An investigational inhibitor as well as an FDA-approved therapeutic. And the patient was responding to treatment, and their outcome measure was their PSA level, because it correlated really well to their tumor burden. The problem was the toxicity was so high that the patient was considering quitting the trial. And by nature of being a trial, this patient had already failed previous treatments. What happened was we were able to get the approvals to obtain the data for this patient. And this is, again, not modeling. This is actual data from the patient of the different doses that they were exposed to. This is an important part of the calibration platform. We need different dose response of this patient in order to calibrate this smooth surface. This smooth surface, which you saw with transplant, which you see with this particular patient, was previously discovered using neural nets on cell lines, on animals, et cetera. And we're able to show that now by dramatically cutting down on the number of data points, we can personalize these profiles, and we can immediately capture how to optimally dose patients. If you take a bird's eye view of this plot here, what it shows us is that at 24 milligrams of this inhibitor, we can decrease the PSA level. The challenge was this patient was originally in the 48 milligram cohort. So this is a 50% dose reduction, but to increase efficacy. And after the necessary approvals, within a week, the patient had the lowest PSA levels they had seen during the course of the entire study. That single data point is put into this profile. And it told us to continue low dosing. And at that additional low dose, the PSA went down even more. Now, during the course of treatment, we would increase a little bit, decrease a little bit, increase a little bit, and beyond. And what had happened there was this dynamic dosing approach led to sustained optimization of patient care. This is very different than standard high dose, where you give a high dose and either the patient responds or they don't respond, right? In another recent study, we had a patient, and this is an important one, was not responding to treatment. We're given a high dose, immediately no response. The cancer markers kept going up, and the standard approach is to move them to another line of treatment. However, we had calibrated this patient, and we were able to get a surface plotted out, and by reducing the dose of one of the drugs, the patient switched from being a non-responder to a responder. And this goes all the way back to the beginning of the talk, where I mentioned even good drugs given at the wrong dose can result in no efficacy. In this particular patient, the team originally thought that this person would not be able to respond to this treatment, when in fact, just by changing the dose, but keeping the drugs the same, they could in fact respond. This is a substantial opportunity to capture more responders. Our goal is not to low dose every single oncology patient. Some patients will need the max tolerated doses. But in this particular case, if we can find more patients that can respond, we can reacquire or acquire high quality of life while maintaining efficacy for those patients. I show this, I'm not going to go into detail, because I mentioned at the beginning of the talk that it's beyond technology. We have to look at how scalable, how sustainable are these approaches. And so we've done parallel studies to look at how much cost savings we are presenting to the patient. So this is for a different patient who had gastric cancer. And what we were finding is that we were saving the patient thousands of dollars a month just on reduced drug cost. This doesn't include the potential for reduced readmissions due to toxicity and adverse events. And so what we're looking at now here is reducing the cost of care for the patient, potentially reducing the cost of care for the healthcare system, but delivering high quality of life and better outcomes. So this is an important element of truly impacting both patients, their caregivers, as well as healthcare systems. Moving on. As we all know, we are currently amidst a pandemic. In November of 2019, we convened a team to look at our platform to optimize how we rapidly design combination therapies for infectious diseases. And in November of 2019, we didn't know about COVID yet. And what happened was, if we look at this, this paper up here, this team expands well beyond engineers and clinicians and scientists. We have global health security and surveillance experts. We had KPMG come in to help with some healthcare econ analysis, and the business school. And what we ultimately showed with this platform, known as Identify, was that we could optimize very rapidly the design of combination therapies against infectious diseases. Now, at this time, we were using VSV. We didn't have SARS-CoV-2. But a few months later, we were confronted with SARS-CoV-2, and working with an amazing team of collaborators here in Singapore, we were able to validate this platform against SARS-CoV-2, the live virus. Similar to the previous study, we do not use pre-existing data to feed algorithms to make predictions about what drugs could be useful. We use actual prospectively acquired calibration, dosing calibration with patients through clinical trials, or when it comes to live virus, we study multiple permutations of this massive combinatorial space to truly optimize the combos, and we rank these combinations out by efficacy and tox. So this is not in silico, it's not modeling driven, this is driven by actual data. And our studies and our collaborators, we have shown that even in vitro optimized combinations have been able to hold steady and hold true all the way into patient care, right? So this is very different than standard dose response predictions of whether drugs will work. This is clinically actionable optimization, but AI-based, right? That correlation, that smooth surface, that's an AI-discovered correlation, but we have to use prospectively acquired data in order to get this information. And so about a couple months ago, we reported a new study with Identify 2.0, a newer set of therapies in collaboration with colleagues at the National University Hospital, NCID, and many other wonderful collaborators, including at DSO, to optimize a new set of therapeutics. But this time around, we wanted to look at therapeutics that were tablet-based, so that there's a potential to provide community deployable therapeutic combinations. And what was cool about this is that we were able to validate these combinations in both the beta and the Delta variants. So it took a wonderful community to come together. But I wanted to point out that this Identify platform is a supportive platform and it supports what is a primary objective of vaccination, contact tracing, and detection. Right. So in the event therapeutics are needed, this platform can rapidly, within a couple of weeks, pinpoint not only which combinations could be effective to pursue, but also what combinations we should potentially avoid. Because it's a ranked list, and so it provides actionable answers. And what's cool about this is our results from this have largely aligned with clinical outcomes, but without requiring any data from these clinical trials. So it's an independent alignment with clinical outcomes. And again, that's because we are using an optimization approach, not just dose response. I wanted to end the ID portion with just a fun message here that, technology alone can't change healthcare. We know this, and we've had the privilege to work with a very multidisciplinary community. And part of that includes education as well. And so we actually started this comic book about 3 years ago. It's Aiden and Eileen versus Vinny the virus. And what you see here is just free download. And it took a massive community to put this comic book together. So for anybody interested, visit the N-of-1 website, and I think it's right on the homepage. Please feel free, a free download of a cool comic book that we made. Final part of the talk. So what I've shown you largely pertains to therapeutic development and dosing. But we're now also looking at digital therapeutics, what we call medicine without the meds, or medicine without the pill. And this is using software as treatment. And we're doing this to look at things like cognitive training, to prevent cognitive decline, and not only for seniors or for healthy aging, but also for oncology patients. And so this is a publicly available platform, with an acknowledgement to Matt B. And what we've done is this is a multitasking platform. And working with radiation oncologists, working with Chris Osplund, what we have is an opportunity to personalize how we used software as an intervention. And so we conducted a study a couple of years ago to show, again, just how individualized participants respond to treatment. And we're using these profiles where we dynamically change. Instead of dose, we change intensity of the gaming of the different modules to try to optimize how people cognitively perform. Right. And so what we have going on here is we've since modernized this approach through wonderful work from Dr. Alex Remus and Jason and Nguyen and others from our team. And so we are now stepping up partnerships and co-creation opportunities with community centers. And we've also recently just initiated a clinical trial for neuro-oncology patients. And so what I've shown you today is kind of this spectrum of the work that we do. We are using AI-based approaches, small data approaches to optimize treatment. And what we've shown is that we do partner substantially with big data researchers to help us select which therapies we should consider in the first place, how to potentially match patients to the right therapeutic pool using genomics. But after we think about the drugs that we wanted to use, we then have to optimize combinations comprised of these therapeutics and then ultimately optimize the dosing. But in order to do that, we use smaller sets of data, and that data is carefully acquired and calibrated to ensure that we are optimally interrogating that drug dose space. The final thing I'll say is that WisDM and N-of-1 and biomedical engineering and pharmacology, our mandate is to transform the way that medicine is practiced. And so everything I've shown you today has demonstrated that we can take ideas, we can take tech, and we can have accessibility to a very large community of stakeholders to show that we can bring these technologies and impact patient care. Everything we do is with the mindset of impacting patient care. And in many cases, we've achieved life-saving outcomes. And we are grateful for the opportunity to be a part of this ecosystem because these life-saving advances are driven by accessibility. Accessibility to stakeholders who can quickly and actionably act on many of these workflows and protocols. And so by teaming up, we've seen really cool and amazing outcomes, and ultimately we've helped some patients out. So I wanted to acknowledge the amazing community that makes this happen and thank the funding agencies that support our work. Thanks for having me today.